Hello, my name is Anna Blakney, and I'm an assistant professor at the University of British Columbia in the Michael Smith Laboratories and the School of Biomedical Engineering. Our goal with any vaccine is to train your immune system to recognize a foreign pathogen without actually being infected by this pathogen. We do this by introducing a piece of the pathogen, a small protein, usually from the surface, which is called the antigen. So there's many ways that we can introduce this antigen into your body, which we can do, typically with an activated viral vaccine or the protein itself, or more recently by using an mRNA vaccine. An mRNA vaccine works by entering your cells through endocytosis and being released into the cytoplasm through an endosomal release mechanism that we'll get into later. Once the RNA is in the cytoplasm, it engages with the ribosome and the antigen is translated, which then goes on to be processed by your immune cells. So there's a couple of different ways that RNA induces an immune response. The first is through innate immunity. So because our bodies have evolved to recognize foreign RNA, as lots of viruses are made of RNA, a typical mRNA vaccine is recognized in the same way, which is referred to as the interferon response. So because of this, mRNA vaccines are considered to be self-adjuvanting, whereas they raise these alarm bells without adding anything else into the vaccine. So this is what triggers what's called your innate antiviral response. But in addition to this, mRNA also triggers your adaptive immunity, which comes in kind of three different flavors. So your CD4 T cells, which works by helping secrete cytokines that support cell-mediated and humoral immune responses. It also invokes CD8 positive T cells, which are responsible for killing infected cells by cytotoxic mechanisms, as well as B cells, which are responsible for antibody secretion. There's a number of advantages and disadvantages that messenger RNA vaccines have compared to other vaccine platforms. So some of the advantages include rapid development of modified versions. So once we have an mRNA vaccine, it's very modular and easy to make a new one, just like we did at the beginning of the pandemic. It's also optimal for antigen expression. So we're able to express just the protein that we want and get a lot of that protein expressed, which is very good for mounting a good immune response. Another advantage is that it's also able to elicit both humoral and cellular adaptive immunity, which is a good way of training your body to recognize these pathogens. And it doesn't matter which immune mechanism you're going for, you get both the humoral and cellular immunity. It doesn't require a live pathogen, so it's much safer to produce on a large scale, and it has this self-adjuvant effect. So we don't need to add anything else into the vaccine to get this training and triggering of your immune system. However, there are also a number of disadvantages to mRNA vaccines. So there can be severe reactions to the polyethylene glycol component. There's also a potential risk of myocarditis in selected groups, and as for now, there is a requirement of cold chain transportation and storage. So they have to be stored in freezers, and this can impede their distribution worldwide. So there's a number of very important components for mRNA. So I'll start with the 5' cap. This is important for increasing the translation of the mRNA. It regulates processing. It shields the mRNA from innate immunity, as well as protects it from exonucleases. At the end of the mRNA, it has a 3' poly A tail, which also helps with the mRNA stability. So this stabilizes and promotes translational efficiency. And we've also found that the tail length of this poly A tail is proportional to the efficiency of translation. There's also an untranslated region at both the 5' and 3' end of the mRNA. This helps with ribosome recognition, recruitment of the translational machinery, and structural folding. Finally, with the open reading frame, this is where we actually put our antigen or protein. This is the coding region for the vaccine sequence, and the codon modifications of this is very important. So this helps with the translational efficiency, the mRNA abundance, the protein folding, and the antigen stability and immunogenicity. In our typical mRNAs, we also use modified nucleotides, which helps reduce the recognition of the innate immune system to mRNA. So another great aspect of mRNA vaccines is the flexibility of the design. So if we think back to the original designs of the COVID mRNA vaccines, it's very simple to just make some small changes in the code and then produce those in exactly the same way that we made the original mRNA vaccine. So it's a very modular platform. We also have some new technologies coming online now, such as self-amplifying RNA, which is kind of a next generation messenger RNA. saRNA has very similar components to a messenger RNA, except that it also encodes a viral replicase. So this replicase allows the RNA to make exact copies of itself once it gets into a cell. And because of this, you get a much longer duration of antigen expression. But another advantage is that we may be able to use a lower dose of mRNA if we use an saRNA compared to an mRNA. One of the main challenges with mRNA vaccines is getting the RNA into cells. mRNA is very susceptible to degradation, and it's a large negatively charged molecule, so it doesn't get taken up into our cells spontaneously. So scientists have explored a number of different delivery strategies to get RNA into cells, such as polymer-based, peptide-based, virus-like replicon particles, and cationic nanoemulsions. But the leading delivery technology in the field is called lipid nanoparticles. So lipid nanoparticles are a nano-sized vesicle that is able to get the RNA past the cell membrane. It encapsulates the RNA completely in the particle and is composed of 4 different lipids that each have a different function. So the first and probably the most important is the ionizable lipid. So it's charged at a low pH, but neutral at a neutral pH. And so what this allows us to do is to formulate the RNA inside the particle and then bring it back to a neutral pH for the actual administration. The ionizable lipid is an essential component. It mediates the mRNA binding with the core of the lipid nanoparticle, and it facilitates endosomal escape. So once the lipid nanoparticle gets into the endosome matures and the pH drops. And as this happens, the ionizable lipid again becomes charged and releases the RNA from the endosome. There's also helper lipids in the LNP. This helps support the lipid bilayer. It promotes endosomal fusion and can also determine the target organ specificity. Another component is cholesterol, which confers the LNP stability. It also promotes endosomal fusion and aids the vaccine complex uptake. Finally, we have the pegylated lipid, which helps to reduce LNP aggregation. It minimizes the nonspecific uptake by immune cells, and it also determines the circulation rate and immune cell uptake. There are also potentially side effects that are associated directly with the lipid nanoparticles. So this includes pain, swelling, fever, and a systemic inflammatory response. I think it's an incredibly exciting time to be in the field of mRNA technology, but one of the most promising applications for me is using mRNA for personalized cancer vaccines, wherein a doctor can go in, take a sample of a patient's tumor, sequence that, and see what proteins are on the surface of it and may be immunogenic, and then prepare a personalized mRNA vaccine that trains your immune system to attack that specific cancer. So because mRNA vaccines are so modular, it is feasible to do this on a per patient basis. And I think this is one of the areas where mRNA can really make an impact.
Hello, my name is Oliver Cornely. I'm a professor and the director of the Institute of Translational Research and also the scientific director of the Clinical Trials Center, CTCC, here at the University of Cologne in Germany. One big advantage of the mRNA platform-based vaccines is that they can be adapted. So while we originally, late in 2020, had 2 vaccines, and both were directed against the wild type, what we today call the ancestral strain of SARS-CoV-2. In the meantime, there have been several changes, and we know that the original vaccines lost protection or the activity against the virus. So what we then saw was that rapid adaptation was possible, and we got bivalent vaccines against that original virus and against the Omicron variant of concern, BA.1. And then on top, after that BA.1 wave, when others became more prevalent, there were new vaccines, BA.4 and BA.5 directed, and recently a monovalent vaccine directed against XBB 1.5. So the most recent iteration, I think we are not at the end of that procedure and that evolution. So what is being done is that the mRNA composition is being changed, and that then induces immunity directed against the most recent strains and the most prevalent strains, and those that raise the most concern. So what was found with mRNA vaccine? The efficacy perfectly outweighed the safety issues that there may be, the safety concerns. So one aspect always with everything is hypersensitivity, right? So there can always be hypersensitivity to either the active substance or excipients that come with the vaccines. The frequently or most frequently reported adverse events are, and that would be the category with like 10% or even more, is, of course, we all know that, injection site pain, swelling, erythema, etc. But that is something that we see with many vaccines, and it's not a surprise because, I mean, you inject into that muscle, so there should be some reaction, ideally. If there is no reaction, and that is rather a concern, if there is a reaction, I tell my patients, well, okay, be happy because your immune system is now busy, and that is exactly what we wanted to do. But there are other symptoms that are more a concern, but they usually go away after at least the maximum of a few days. That would be a fatigue or a headache that might happen, or myalgia, chills. So it's all in the range of what we are used to with other vaccines, and of course with infections, right? So the warnings and precautions are in the direction of an elevated risk of myocarditis and pericarditis, specifically in young male vaccinees. So that is something to keep in mind. Usually that happens during the first 2 weeks after injection, and it's more common after the second dose. So, but the risk as such, the absolute risk, so how frequent is that? How frequent do I face an event? Well, with these numbers that we are looking at, we can refer to 1 million person years and evaluate how often, how frequent are these AEs. And there are adverse events like ischemic stroke or appendicitis or acute myocardial infarction or that myocarditis, pericarditis, and they are in the range of between 100 and 2,000 per 1 million person years. You will see stroke, appendicitis, and everything else what I mentioned. However, you will likely not see it more frequently than what you observe in your population that you care for anyway, before you even started vaccination. And one has to keep in mind that the virus infection as such causes all of these adverse effects, all of these diseases, symptoms, and signs of COVID-19 at a much higher frequency than the vaccines do. Thankfully, we can follow the guidance because the guidance always changes with the new pandemic waves or with the new variants of concern. So we should always refer back when we note that there is new guidance from the WHO or from regional or national or local recommendations. And the challenges that we have is, first of all, that the guidance changes pretty frequently as compared to other vaccines where the guidance usually is stable for many years. But with the rapid pace with mRNA platform-based vaccines being adapted to the actual viruses and real diseases that we deal with, we see more changes of the guidance. And what one can say generally, although the guidance in these different bodies from WHO all the way down to regional and local might differ from each other a bit, or might have different days when they are finally being issued, coming to our hands. There are the key updates you should really focus on is whether there are new priority risk groupings and whether there are specific guidance for priority use, specifically if there is not enough vaccine. It's now a rarer issue than it was during the earlier days of the pandemic, but where clearly one had to triage and consider, well, I have only one dose, but 5 people, so who should get that dose? But we are now somewhat more relaxed at that aspect, I would say. Then another point is pregnancy, pregnant women. We all remember that in the old days, pregnancy was seen as a contraindication to vaccination. Then we had to learn that during influenza waves, that was a very bad thing to do because pregnancy is some form of immune suppression and pregnant women are more vulnerable. So the current recommendation is to definitely not withhold the vaccine in pregnancy. Ideally, one would give the vaccine in the second trimester, and that is because you want to give it as early as possible, but we all always avoid the first trimester for obvious and known reasons. So that is why it's the second trimester. And then other recommendations where you see differences between the general population and healthcare workers. So healthcare workers should have another booster every 12 months. And that healthy children and adolescents, we may see some change there because there are some ongoing clinical trials, but currently they are being considered for primary series, and that differs from country to country actually. And it depends on the context, disease burden, cost, and other aspects. With all of these numbers, like healthcare professionals give them another shot every 12 months, you always would need to factor in whether they had a infection with coronavirus during the last 12 months, because that would count as well. And if that was the case, then you could actually add another 12 months to that last exposure. With the advent of a new platform, mRNA vaccines, and we all witnessing that they can be adapted and can react so rapidly. So now there is, of course, great enthusiasm, and I'm not an exception. I'm enthusiastic about these too. And I'm, of course, dreaming of, what all could be done with these. And I mean, we know that we are looking for a vaccine against HIV for ages. And while many patients with HIV are now living with drug treatment, with chronic drug treatment, and they do, of course, much better than in the 1980s. Still, they have that chronic infection. They don't get rid of it, and it would be fantastic to have a therapeutic vaccine or a vaccine before infection. Both aspects are being worked on right now, as I see from the literature, but there are many, many others. So influenza, for example, is a pathogen of pandemic potential. Clearly, we know that it caused, and influenza A specifically caused pandemics in the past. So we would love to have a better vaccine than the current 3 or quadrivalent vaccines that we use. Parainfluenza is something that I would love to be able to prevent with vaccines, because I see it in my hematology patients, and it's a terrible disease, and it's, of course, easily transmittable. And there are other other pathogens, rhinovirus. I mean, maybe not that important, but that would mean quite something if we could avoid rhinovirus infection, because we all have it at least once in a year, I guess. And then there's the whole range of herpes viruses. It's all the human herpes viruses, all 8. But I mean, if we could wipe out HSV 1 and 2, why not? I mean, that would be great, because there is no reason why we should suffer from these two occasionally with when there are these blisters at the lips or in immune-suppressed patients. It can cause a real concern. And then there are other pathogens. Well, one can go down the HHV, the human herpes virus list, to VZV. Well, we have a vaccine, that's cool, but we don't have a vaccine against CMV, against Epstein-Barr virus, and then certainly not against HHV 6 and 7. These are viruses of concern in immune-suppressed patients, as is KSHV, causing Kaposi sarcoma in immune suppression, specifically, of course, then in patients living with HIV and AIDS. So there is a whole lot of viruses, and this list is not comprehensive, but these are my, 15 or so favorite viruses that I would love to prevent with mRNA vaccines. And I know that many of these are under development and even in clinical studies right now.
Hello, my name is Ann Falsey. I'm a professor of medicine at the University of Rochester in Rochester, New York, here in the United States of America. So although RNA vaccines have actually been in development for quite some time, the COVID-19 pandemic has really afforded an opportunity to understand how these vaccines can work and what potential side effects are. So we now know that efficacy has been confirmed in wide range of populations, vulnerable people, various conditions. One of the great things about RNA is how rapidly it can be scaled up, permitting it to adapt to new pathogens, emerging variants. And that's a real advantage when you're dealing with a changing landscape. And as I said, they've now been used in millions and millions of people. And so we really have a good sense of efficacy and safety. And so a few of the caveats that we've learned along the way, there are rare but serious side effects. The main being the myocarditis, pericarditis, that it's seen mostly in young men. And that's one thing associated with the RNA vaccines. Fortunately, it seems to be relatively mild, and there have not been any deaths directly attributed to it. There does seem to be a limited duration of protection against infection, although I will say that the duration of protection against severe disease is quite good. We've struggled in COVID with reduced efficacy when we have new and emerging variants, and that can be an advantage of the RNA, that you can, again, switch it up pretty quickly. One of the logistical problems with RNA vaccines is thermostability and ultra-cold storage requirements, and I think that presents some issues with equity in resource-poor settings. So we learned a lot with the mRNA vaccines with COVID, and so now there's a fair amount of enthusiasm to use this technology against other viruses and, in particular, respiratory pathogens. One of the programs to use this technology is against Epstein-Barr virus, and that is the cause of mononucleosis and could cause a fair amount of illness. And that program is moving along, and studies are being done. One that's fairly advanced is against RSV, known as respiratory syncytial virus. And that's a respiratory virus that causes severe illness in very young babies and in older and frail adults with underlying heart and lung diseases. The top-line results of a phase 3 study are in, and it looks quite good and effective against lower respiratory tract illness. And then one that's very appealing is using this technology for flu. And flu vaccines contain 4 different strains of flu virus. And as we know, flu is very changeable, and authorities have to guess each year which is going to be the circulating strain. The older technology with the protein vaccines was you needed a fair amount of lead time, but RNA vaccines are very quickly adaptable should a new strain appear on the scene. And so we are looking forward to seeing the results of those studies. And then lastly, there are some combination studies. So there's a myriad of respiratory pathogens. One is human metapneumovirus, and then there are the parainfluenza viruses. So there's a program combining human metapneumovirus and parainfluenza 3. So even though the RNA vaccines are quite good, there are some improvements that might be considered. And one of the techniques is what's called self-amplifying RNA. And that's where the RNA is combined with what's called a replicase. So it's got embedded something that will lead the RNA to replicate itself in the cell. And what that might do is allow sort of a more prolonged stimulation of the immune system and potentially better duration. And it's also possible that you could use lower doses of the vaccine. And that may be an advantage. As I mentioned before, there's the potential to do multivalent vaccines. That is, combine stimulate immunity to several different pathogens. And this could be actually different viruses, like I mentioned with human metapneumovirus and parainfluenza, but it also could be against different strains or variants of a single virus. So, what comes to mind there is COVID. That it's been very hard. We've been chasing different variants, and so you could try to design a sort of universal vaccine incorporating the major lineages of a single virus. I think one important thing that we have to work on is improvements in storage requirements. Something like freeze drying that would allow storage at higher temperatures for a more prolonged period of time. And this would go a long way to having it be more widely available globally and global equity. And then there's novel routes of administration. One can give an intranasal delivery. And what is hoped with that approach is that you might actually stimulate mucosal or nasal antibody, and it would actually induce what we call sterilizing immunity. That is, you don't get infected at all, and then you can't transmit it to others. What we have right now is it reduces symptomatic illness and particularly severe illness, but maybe not optimal for preventing any asymptomatic infection. So the RNA technology for vaccines is really exciting in many ways, and it's hard to choose one most important feature. I think that we always knew about the threat of global pandemics. And, theoretical is one thing, and living through a pandemic is quite another thing. And so we really need vaccines that can be developed and scaled up really quickly. And now we have that with RNA. And so the potential for pandemic vaccines is enormous. And, unfortunately, I think they are in our future, hopefully not for quite some time. But the other thing that I think is pretty exciting is the ability to have multivalent vaccines and combine different strains and different pathogens. As we have more adult vaccines, you don't want people to feel like pincushions. And if you can incorporate multiple pathogens, that would just be really terrific. Adult RSV for many years has been underappreciated and underrecognized, but there's now very clear epidemiologic evidence that it is a significant cause of morbidity and mortality in older adults. Just as an example, in the United States, there's roughly 100 to 180,000 hospitalizations and somewhere between 10 and 15,000 deaths, which can be attributed to RSV. The people that are most at risk are those with advanced age and then individuals with underlying heart and lung conditions. And so having a vaccine that can actually prevent illness, and especially hospitalizations and deaths, would be very impactful. I think one of the things that's also underappreciated is that it is a pathogen in long-term care facilities or nursing homes, and having a vaccine for our most frail elderly, again, could be very impactful. The RNA vaccines have shown to be very immunogenic and I think would be quite safe and well tolerated. The COVID vaccines were very well adopted by the long-term care facility medical directors, and so I would hope that if we can develop a successful RSV vaccine, that would be a population that would be very important.